Welcome back to MedTechISO. I am your host Arvind Rathore, and in today's video, we are exploring one of the foundational steps in ISO 109931 compliance: chemical characterization. If you have ever wondered why this step is so critical or how to carry out it effectively, this video is for you. We will discuss the role of chemical characterization in ensuring device safety, the analytical techniques used to gather data, and how to report your findings to meet regulatory expectations. Let us get started. Let us first discuss the role of chemical characterization in ISO 109931. Chemical characterization is the process of identifying and quantifying the chemical substances present in a medical device. This step forms the basis of biological safety evaluation because it helps predict how a device might interact with the human body. Simply put, you can't evaluate what you don't know. Before you move on to the biological testing, you must first understand what chemicals are in your device, how they might leach out, and what levels they could be harmful. Why is this step so important? Chemical characterization acts as a gateway to subsequent toxicological risk assessments and biological evaluations. By identifying the chemicals released by your device, you can determine whether they pose risks such as toxicity, irritation, or sensitization. This step is particularly crucial for devices with prolonged or permanent contact with the body, like implants, or even trace amounts of harmful chemicals can cause long term health effects. Another critical role of chemical characterization is in demonstrating regulatory compliance. Regulatory compliance bodies like the FDA and EU MDR require manufacturers to provide detailed chemical characterization data to support their safety claims. A thorough chemical characterization study can also help reduce the need for extensive biological testing by showing that certain risks are negligible based on the chemical profile of the device. Let us now discuss how chemical characterization fits into the ISO 109931 framework. Chemical characterization is typically performed as a part of the broader risk management process outlined in ISO 109931. The data collected during this step informs toxicological risk assessments and help you decide which biological tests are necessary. For example, if a device releases a known irritant, irritation testing becomes essential. If a device shows no significant leachable or extractables, certain biological tests may be waived with proper justification. It is important to note that chemical characterization doesn't just evaluate the raw material used in the device, it also considers additives, substances like plasticizers, colorants, or stabilizers, manufacturing residues, chemicals induced during production, such as lubricants or cleaning agents, sterilization byproducts, residues from sterilization methods like ethylene oxide or gamma irradiation, degradation products, substances formed when the material breaks down over time in the body. Each of these factors can significantly impact the safety of your device, making chemical characterization an indispensable step in ISO 109931 compliance. Next, let us discuss the analytical techniques for chemical characterization. The success of chemical characterization relies heavily on the analytical techniques used to identify and quantify chemical substances. ISO 1099-318 provides guidance on how to perform chemical characterization, including the selection of extraction conditions and analytical methods. Here are some of the most commonly used techniques. Number one is gas chromatography, mass spectrometry (GCMS). This technique is ideal for detecting volatile and semi-volatile organic compounds. For example, GCMS can identify residual solvents or low molecular weight additives in polymer. Number two is high performance liquid chromatography (HPLC). HPLC is used for non-volatile compounds such as plasticizers or leachables that dissolve in body fluids. It is particularly effective for identifying polar and high molecular weight substances. Number third, inductively coupled plasma mass spectrometry (ICP-MS). ICP-MS is the go-to technique for detecting and quantifying metals and trace elements. This is especially important for devices containing metallic components or coatings. Number fourth is Fourier transform infrared spectroscopy FTIR. FTIR is used for identifying specific chemical bonds and functional groups in materials. While it doesn't provide quantitative data, it is useful for material identification. Number fifth is thermal analysis TGA and DSC. Thermal techniques like thermogravimetric analysis TGA and differential scanning calorimetry DSC help understand the stability of the material and detect potential degradation products. Number six is nuclear magnetic resonance spectroscopy NMR. NMR provides detailed structural information about the organic compound and can help identify complex molecules that other techniques might miss. The choice of technique depends on the device's material, expected leachables, and the conditions of use. Often, a combination of these methods is needed to create a complete chemical profile of the device. Reporting chemical characterization data. So, now let us discuss the reporting of chemical characterization data. Once you have completed your chemical characterization, the next step is to compile the findings into a comprehensive report. This report will be reviewed by the regulatory bodies, so it needs to be detailed, accurate, and well organized. Here is what your report should include. The first is device description. Provide an overview of the device, including the materials, manufacturing processes, and intended use. Highlight the components that were evaluated during chemical characterization. Number two is extraction conditions. Explain the extraction methods used to simulate real world exposure conditions. For example, did you use saline or the ethanol to mimic body fluid? Did you heat or agitate the samples to accelerate leaching? Be specific. Number third is analytical methods. List the techniques used for analysis such as GCMS or HPLC. Include details about the equipment, calibration, and detection limits to demonstrate the reliability of your data. Number fourth is chemical profile. Present the results of your analysis, including a list of all identified substances, their concentrations, and their toxicological significance. Use tables and graphs to make the data easier to interpret. Number fifth is risk assessment link. Connect your findings to the toxicological risk assessment. For each substance identified, explain whether it poses a risk based on its concentration and toxicological profile. Number six is conclusion and recommendation. Summarize your findings and discuss any actions taken to address identified risks. For example, if a substance exceeded safety threshold, explain how you mitigated the risk. A well-prepared report not only satisfies regulatory requirements but also provides a strong foundation for subsequent evaluations. Remember, clarity and transparency are the key. Regulators should be able to understand and verify your findings without additional clarification. So, in conclusion, to summarize, chemical characterization is a critical step in ISO 10931 compliance because it provides the data needed to evaluate the device's safety. It identifies potential risks from materials, additives, and byproducts, forming the basis for toxicological and biological testing. Using advanced analytical techniques like GCMS, HPLC, and ICP-MS ensures that your data is accurate and comprehensive. Finally, a well documented chemical characterization report ties everything together, demonstrating your commitment to patient safety and regulatory compliance. If you found this video helpful, please like, share, and subscribe to MedTechISO. Do you have questions about chemical characterization or need advice on selecting analytical techniques? Drop your question in the comments below. I am here to help. Thank you for watching, and I will see you in the next video as we continue our journey through ISO standards for medical device safety.